I will move on now with Stéphane Ralite. Stéphane is the Aquaculture Product Manager of Lallemand. He has a solid experience and knowledge of the aquaculture segment in different regions of the globe. He has raised shrimp in Thailand and held several positions within Evialis in Vivo Group in France, playing a role in the setup of its aqua division in Ocealis. After being aquaculture market manager with Diana Group for its Aquatype division, he set up his own consulting company, bring his aqua expertise to different organizations. In 2014, he is in charge of the global development of Lallemand Animal Nutrition aquaculture activity worldwide. He'll be speaking today about the value of functional yeast in aquaculture feed production. So I will hand over to you, Stéphane.
Okay, thank you very much, and hello everybody. So I will start also by introducing a very quick introduction of Lallemand company. Lallemand is an old company because it has been founded more than centuries ago. We have over 5,500 employees, and we are specialized in yeast and bacteria. We produced only yeast and bacteria. This is our plants, and you see 29 plants which are doing fermentation. We are primary producer, primary fermenter of yeast and bacteria. And we sell the yeast and bacteria in all the market which are able to interested by live yeast and live bacteria or its extract. Aquaculture is just one part of the company. It's a part of the animal nutrition, but we are also working on the plant care, the human health, human food business, biofuel production, etc. So yeast, it's a natural microorganism. It's actually, it has been part of the human diet for a long time, as Egyptian was already fermenting wine and doing some leavened bread. But in the recent years, we have seen increasing awareness about the nutritive value of yeast, and it's interested also for health. So the yeast cell wall, it is a eukaryote cell, which is surrounded by membrane, thick membrane, which contains some specific compounds like MOS, mannan protein, which are on top of the beta-glucan, as you can see here. The beta-glucans itself surrounded the thin chitin layers, which protect the cell membrane, the phospholipid membrane. Inside the cell, you have a lot of components also, like amino acid, peptides, protein, minerals, and sometimes organic minerals, or enzyme, vitamins, and some compounds which are known as nucleotides, so in different forms. I will not go into details here. So these terms like nucleotide, mannan, MOS, glucans are well known. It has been demonstrated to be efficient, and we've proven efficacy for many years now. But in the recent years, we have seen more and more company and more product arriving on the market. And it's sometimes difficult to compare the different products. So I wanted to give a simple classification and some simple guidelines to help sort the different yeast products that are available in the animal nutrition market, and yes, give some guidelines to help understanding the difference between them and help to choose. So as it has been already stated, thank you, Derek, the first difference is it live or not? The probiotic, it's a live cell, which is active in the gut of the animal, and it confers some specificity. As it is live, it is integrated as inside the microbiota, and it has a real metabolic activity and reactive activity depending on the conditions. If it's not live, then we have a lot of different non-live yeast families. The first one are what I call the yeast culture. So there is a different kind of yeast culture. You have the yeast, which is complete, not processed, and with part of the media that serve to ferment the yeast. So inside the media, you can have some metabolites. So depending on the process, depending on the selection and the efficacy of the fermentation, etc., you can have more or less metabolites and more or less variability inside the product. Then a second family of product is what I call the inactivated yeast. So inactivated yeast is just dead yeast. You have the full yeast and with the full membrane, which trap, let's say, the components of the cell. Then you have some autolyzed yeast. So the autolyzed yeast is a yeast which is dead, and that where the endogenous enzyme from the cell start to lysis the cell, and especially the cell's membrane. Then you have some more specific possible lysis with the addition of exogenous enzyme. This is what I call the hydrolyzed yeast. And then, of course, it's much more specific because you can choose the enzyme depending on the kind of hydrolysis that you want to do, where you want to cut the molecules, etc., and then to target some specific production of specific molecules, etc. Then you have a big family of product. When you inactivate, when you hydrolyze, and you can separate the different components, what I call yeast fraction. So you can separate the yeast cell wall from the cytoplasm, and inside the yeast cell wall, you can also break the yeast cell wall to extract the specific components that you want to get. So this is the yeast fraction family, where you can have some moss, some beta-glucan, some nucleotides, specific and rich product. And you can also formulate the different parts. And then you can enrich the yeast with some specific components like minerals or vitamin D, for example, or a very well-known also a product on the market, which is organic yeast selenium. So it's the same, depending on the process, you can have 100% organic selenium present inside the yeast. Another important parameter to consider is the origin of the yeast. Is it a primary fermentation yeast? Primary fermentation, it means that you select the yeast specifically for the final utilization that you are going to do in the feed. So you select the yeast and you select also the process, especially the fermentation process, to maximize the impact that you want to get in terms of immunity, for example, or pathogen binding, etc. The other kind of product is the secondary fermentation. So it is a byproduct. So it means that the yeast is used to, for example, ferment a beer or to produce some biofuel. And then you use the yeast afterwards to develop some other products. So the function of yeast that are usually used are here. The characteristics are here listed in the left of the sheet. So I will not detail everything because I will not have time. But yeast are a source of protein, but you have to check the digestibility of this protein. So here, for example, for the inactivated yeast, it contains protein, but the problem that it's trapped inside the cell wall, so the digestibility is not optimized. On the contrary, if you do some specific hydrolysis, you can increase a lot the digestibility. Same for the functionality, like immune enhancement, pathogen binding, gut health, skin mucus protection. I will give some more example. Depending on the process and the origin of the yeast, you can, for example, once again, in the basic inactivated yeast, the functionality will be very low because the cell wall is just still complete and not, and the molecules like the beta-glucans, for example, are not exposed at all. On the contrary, once again, if you look to the yeast fraction, which are specifically processed and selected for this, you will have much more, of course, efficacy, functionality. So let's take one example, which is yeast cell wall. What are the possible benefits of yeast cell wall? It is well known for some times already. The mannan oligosaccharide, for example, are able to block undesirable bacteria, and then they attach to these bacteria, and the bacteria are not able to attach to the cell host, the fish or the shrimp cell. Another very important function is, for example, the immune enhancement response. So with beta-glucan, it is also quite known, you can stimulate the immune cells through specific receptors that will enhance and increase the immune response. If you support the immunity, you can also have some impact on the mucosal barrier, for example. So you can increase and play on the mucus quantity and the mucus quality of the fish, or inside the mucus also, and the immunity inside the gut. So this is, you have different components in the yeast cell wall, we will see, that can influence the immunity. And then you have the prebiotic effect, which is also well known by just favoring some bacteria like the lactic acid bacteria. So are all yeast cell wall alike? No. You understood that, for example, depending on the origin, is it from primary fermentation or is it a byproduct, then the composition and the efficacy will be different. This is one example when you just analyze the basic composition in mannan and beta-glucan from some byproduct present compared to the primary fermentation group, you see that the composition is different. You can also check some basic particle size. We checked some different MOS product claiming some MOS activity, and then you see the particle size in the different product are very different. You can have some big particles and some very small particles, which will influence especially the binding capacity. So this is simple analysis that you can do to start sorting the different yeast cell wall yeast products that are present and proposed on the market. But on our side, we wanted to go further and to better understand the efficacy, the function, and the ability, for example, to do pathogen binding or immune stimulation. We wanted to better understand the structure of the yeast and the influence of this structure on the functionality. And thanks to new technologies, we were able to go much further and to screen different strains. One of these new technology is AFM, so which is atomic force microscopy, which allow to look at the yeast at the cell level. So we are able to analyze the surface of the yeast cell wall and to check, for example, here you see the 3 dimension of the yeast cell wall, which will have an impact on the function and the efficacy. Or we can select to look at specific molecules, for example, some binding molecules, and to analyze these molecules in terms of 3 dimension, in terms of structure, in terms of strength, for example, etc. And to link these structure to the binding property, the pathogen binding or the impact on toxin, mycotoxin, for example, etc. And then this allows us to do much more screening and to use all our strain that we have. We have some huge bank of strain, and to screen all the strains and to select the strain depending on the function that we want to develop with the product. And this leads to what we call new generation yeast fraction. And here one of them is the young product, which is a mix, a formulation of different part of a different yeast strain. So here you see that you have two Saccharomyces strain and one new species, which is Ciberningera. And we select also different part of the yeast to maximize. So to maximize what? To maximize pathogen binding. So here, this is an example of one screening that we do for aquaculture and especially for shrimp. You see that we check the ability to bind, to attach Vibrio, 3 different strains of Vibrio paramelliticus. And here it's Escherichia coli, it's a positive control that we know well. And we compare the standard yeast to the 3 different fractions that has been taken to make young. And you see that minimum, we double the binding capacity here. With this fraction, we are even at 100%. So here, and you see some yeast with, so here it's a picture of Escherichia coli, but which is glued to the yeast, attached to the yeast. Another function that we wanted to develop is the immune modulation. So we select also the different yeast. The objective was to maximize the immune response. What is well known is that beta-glucan, for example, has the capacity to induce some immune stimulation, and it stimulates the immune cells through one major route, which is through dectin-1 receptor, which is a specific receptor present on the immune cells. So the beta-glucan will attach and stimulate the response. We know that yeast cell wall in general can have some influence through other receptors, but our objective was to maximize this response, maximize the ability of the yeast cell wall to stimulate the other receptors to have a much broader immune response, which we had, and here we have a patent pending for this. So what we were able to demonstrate that because of the formulation of the product, by formulating these different components, we were able to have a synergy in the immune response. It means synergy, it means that 1 + 1 is not equal to 2, but equal to 3. So this is here what you see. Another impact also with this much broader stimulation through different receptors allows what we call an immune modulation compared to the immune stimulation. So we can sustain immunity for a longer period without risk of immune fatigue or adverse performance. So practical results, we have done some trial on shrimp with EHP trial. So EHP, it's an intracellular microsporidy. So the only way to help the shrimp to cope with this pathogen is through immunity. And here you see that we were able with young to reduce a lot the number of pathogen inside the cell of the hepatopancreas, which helped to increase the final weight and to reduce a lot the diversity in terms of size at harvest. Another experiment with the MS, here EHPND, so with Vibrio paramelliticus. Here you see that the challenge was very tough. We had only 10% survival in the control, but with the young, we were able to increase a lot the survival, so from 10% to 60%, which is a huge protection. Another example with, I will go quickly with a white feces trial, once again in shrimp, where we were able to protect. So here white feces is impacting the hepatopancreas. And when we score the hepatopancreas, you see that with young, we had much better hepatopancreas. We were able also to reduce the Vibrio content inside the hepatopancreas, or to reduce the growth symptom globally of white feces. For fish, some other example, if you compare moss to beta-glucan to young, and we did some experiment to check the capacity to help wound healing, which is very important in many applications. And you see that young had a much better capacity to improve the wound healing. Or the last example will be on farm. It's a trial that has been done on farm in Japan. The ability to reduce the parasitic load, the skin flux, which is a major problem there on the fish called amberjack. And with young, once again, in the farm, we were able also to reduce the skin flux counts on the fish, which help also to improve the growth and the condition factor of the fish. So this is only a few examples. Thank you very much for your time. And if you have some precise question, I will be happy to answer.
Thank you, Stéphane. There's a lot of information on yeast there today. It's a certainly an amazing organism with there are a lot of beneficial properties to utilization of yeast in various forms.